So ketamine, right. We mostly think about it for pain, maybe sedation, or, increasingly for mental health stuff. But the research just keeps surprising us. Now it looks like it might be this really specific tool against cancer.
It's definitely one of the more unexpected areas in oncology research right now. Taking drugs we already know well and finding they can, well, attack cancer cells in totally new ways. And today, yeah, we're digging into a new study looking right at ketamine and cervical cancer. And it gets really specific, like down to messing with the actual power source inside the cancer cell.
Okay, so that's our mission for you today. We want to understand how this common anesthetic is actually being looked at as maybe a precision weapon against a really tough cancer. We're going to explore this fascinating research by Fang and colleagues from 2025. It gets into exactly how ketamine targets mitochondria, those tiny power plants, and why this is, well, potentially a big deal, even if it's early days.
Right, let's unpack that mechanism.
Okay, so first, maybe just a quick bit on cervical cancer. We know it's really dangerous when it spreads, right? Metastasis causes, what, up to 90% of deaths?
That's right, which makes finding targeted treatments, things that can work systemically, so incredibly important. And this study, like a lot of cancer research now, it really zeroes in on the mitochondria, those cellular power plants.
Yeah, the power plants. Can you give us just a quick refresher? What exactly do they do again?
Sure. Mitochondria are, well, they're fascinating, actually. They're the main energy suppliers. They make ATP, that's the fuel for pretty much everything a cell does. But crucially, they're not just static little blobs. They're super dynamic. They're constantly changing shape, merging together, that's fusion, or splitting apart, which is fission. And cancer cells, they often hijack these dynamics. They need specific mitochondrial states to grow fast and, importantly, resist dying. So messing with that balance, that's a huge goal for researchers.
Got it. So if the cancer cell is like this out-of-control factory, the mitochondria are the generators keeping everything running nonstop. We know hitting those generators is key. But what's new here? What does this study say ketamine specifically does to the mitochondria in these cervical cancer cells?
They use the C33A line, right?
Exactly. The C33A line was the main focus initially. And the core finding, the really unique part, is that ketamine triggers massive mitochondrial fission. And dysfunction comes right along with it. Fission means fragmentation. So instead of those efficient, long power plants, ketamine basically forces them to shatter. They break apart into these little round pieces that just don't work properly.
Whoa, so it's not like a slow drain of power. It's more like flipping a switch that makes the generators just break apart. And that causes this huge energy crisis inside the cancer cell, stopping it dead in its tracks. That's a great way to put it, a massive energy crisis. And this mechanism, it seems designed to push the cell towards apoptosis. That's programmed cell death. When the cell loses its power and that dynamic balance is just gone, it basically triggers its own self-destruct sequence. And like you said, because cervical cancer is so deadly once it spreads, finding these unique, targeted ways to make the cancer cells commit suicide, essentially, is incredibly valuable. We're hitting them right where it hurts their energy supply.
Okay, yeah, that context about metastasis really drives home why finding these vulnerabilities is so important. So if ketamine is causing this massive energy crisis, how did the researchers actually measure that? What did they see happening in the lab with those C33A cells?
Right. They looked really closely after treating the cells. They use a specific dose, 100 micromolar or RENM for 24 hours, and they saw, well, several key signs that the system was failing.
Okay, what were those signs? Lay them out for us.
Okay, first, and maybe most obvious, energy production itself just tanked. ATP levels plummeted. Makes sense, right? Broken generators, no power. Second, they looked at the machinery making the energy. Specifically, complex 4 activity, that's a key part of the mitochondrial electron transport chain. Its activity dropped way down, so the whole assembly line was seizing up.
The energy production line itself just grinds to a halt.
Exactly. And third, they measured something called the mitochondrial membrane potential, or MMP. You can think of that as like the electrical charge across the generator's membrane. It needs that potential difference to work efficiently. When the MMP drops, the whole system basically collapses. And all of this together resulted in clear cytotoxicity cell death. They saw markers increasing that basically leak out when cells are dying.
And this wasn't just a fluke in one specific type of cervical cancer cell.
No, that's crucial. They checked it in 2 other well-known cervical cancer cell lines as well, HeLa cells and CHA cells, and they saw the same thing. That consistency really strengthens the finding. It suggests it's a core mechanism.
Okay, this is super interesting. Now, how does ketamine know to do this? Is it just poisoning the cell generally, or is there something more specific going on, like a molecular switch?
Ah, that's the really elegant part of the study. They actually pinpointed the molecular pathway. It's not random. Ketamine interacts with specific signaling proteins that control how mitochondria behave.
Okay, so who are the key players in this pathway?
Well, the main protein that physically causes the mitochondria to break apart the fission is called DRP1, dynamine-related protein 1. Think of it as the molecular scissors that cut the mitochondria. And what they found was ketamine increases the levels of DRP1, especially its active form, the phosphorylated version, pDRP1, so more active scissors.
Right, so ketamine brings in more active scissors, but what tells the scissors to start cutting? What's upstream?
Exactly the right question. And that's where the signaling comes in. The study found ketamine also boosts other proteins like CAM and K2, and really critically, one called ROOA. ROOA is a small signaling protein, and in this whole process, it seems to act like the ignition key. It's what turns on the DRP1 fragmentation machinery after ketamine shows up.
Wait, hold on. So ROOA is the switch, the thing that tells GRP1 to start chopping up the mitochondria. That level of specificity is kind of mind-blowing.
It really is. And they didn't just observe correlations, they did a fantastic experiment to prove it. They used a specific chemical, Y27632, which is known to inhibit ROOA. When they added this inhibitor, it blocked the whole ketamine effect. No mitochondrial fragmentation, no GRP1 activation, and importantly, no energy dysfunction or cell death. It shut the whole cascade down by blocking ROOA.
Wow. Okay, that's pretty compelling. Blocking the switch prevents the whole chain reaction. Finding that switch seems almost more important than just seeing the fragmentation. Because maybe you could target that switch specifically. What does this mean for, future treatments?
Well, the big implication is it offers a potentially new angle. Instead of broad-spectrum chemo that hits all fast-dividing cells, maybe you could use ketamine or something like it as a mitochondrial dynamic modulator.
Yeah.
The idea would be to specifically target and break the energy system of the cancer cells, hopefully leaving healthy cells mostly alone. It's about hitting a known vulnerability with more precision.
That definitely sounds like a smarter strategy. But, if it's this complex molecular target, does that make it harder to actually turn into a medicine, especially thinking about the dose they used?
Yeah, that's the critical reality check here. We absolutely have to remember this was in vitro, in a petri dish, not in a person, not even in an animal model yet, in this specific study context. And the dose is a big hurdle right now. They used 100 MU. That's quite high. When ketamine is used for anesthesia in patients, the levels in the blood are way lower, usually around, say, 6 to 10 mM.
Okay, so there's a big gap. You can't just give someone an anesthetic dose and expect this mitochondrial shattering to happen in the same way, or safely.
Absolutely not. So the next steps are clear. Translational research, clinical studies. Can these effects be seen in vivo in a living system? Can they happen at lower, safer doses? Maybe by combining ketamine with something else or developing ways to deliver it more directly to the tumor. The finding itself is really promising. It opens a door, but proving it can work safely and effectively in patients, that's still a long road ahead.
Okay, so let's bring this home for you listening. The big takeaway is we can now add cervical cancer to this growing list of cancers, where ketamine shows some potential anti-cancer activity, at least in the lab. The breakthrough here is identifying how it might work by triggering mitochondrial fission, basically shattering the cancer cell's power source, and, crucially, finding that molecular switch, the ROA or DRP1 pathway that controls it.
Right. And maybe the thought to leave you with is this. If a drug we've known for decades, like ketamine, can be found to target something as fundamental as cancer cell energy metabolism with this kind of molecular precision, messing with the ROA pathway to break mitochondria, what else might be out there? What other established drugs, maybe things that are not even used for cancer right now, could have these hidden talents? Could there be simpler ways hiding in plain sight to exploit these core vulnerabilities in cancer cells?
That's a really provocative question. Definitely food for thought. Thanks for helping us unpack the science behind this fascinating research. And thank you, as always, for joining us for this deep dive.